Surrounding the entire muscle is the epimysium. A whole muscle is made up of many fascicles. Every fascicle is encircled by the perimysium. Both the epimysium and perimysium are dense, irregular connective tissue. A fascicle is a bundle of 10 to 100 or more myofibers. Each myofiber has little thread-like structures that extend across the entire fiber called myofibrils. Their prominent striations make the entire skeletal muscle appear striated. A nerve action potential from the central nervous system arrives at the axon terminal, and this causes the calcium channels in the membrane to open up, allowing calcium to move into the synaptic knob. This increase in calcium causes the synaptic vesicles filled with acetylcholine to undergo exocytosis at the axon terminal's plasma membrane in the synaptic knob. During exocytosis, the synaptic vesicles will fuse with the membrane, releasing acetylcholine into the synaptic cleft. The acetylcholine will then diffuse across the synaptic cleft to the motor end plate, where two molecules of acetylcholine will bind to an acetylcholine receptor, causing a sodium-potassium channel to open. Sodium will then travel through the channel into the muscle fiber, making it more positively charged, causing a muscle action potential. Once the nerve action potential stops, the leftover acetylcholine will be broken down into acetic acid and choline, which cannot activate the acetylcholine receptors, by an enzyme called acetylcholinesterase. This action potential will travel along the sarcolemma into the T tubules and will trigger the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it was stored in the calsequestrin, into the sarcoplasm of the muscle cell. Aerobic cellular respiration is a series of oxygen-required reactions that produce ATP, the energy of muscle contraction. It takes place in the mitochondria. This process begins when pyruvic acid enters the mitochondria, where it is completely oxidized in reactions that produce ATP. The calcium from the sarcoplasmic reticulum will then flood over the thin filaments and bind to troponin, causing it to change shape and pull the tropomyosin off of the active sites located on the actin. Now, the thick filaments called myosin will begin to start the process of contraction. Myosin is a golf club-like structure that has a tail and a head. Each myosin head has an adenine triphosphate binding site and an ATPase, which is an enzyme that hydrolyzes adenine triphosphate into adenine diphosphate and a phosphate group. The ADP and phosphate group are still attached to the myosin head. This hydrolysis reaction causes the myosin head to go into high energy state, where it will bind to the active site of actin on the thin filaments, forming a cross bridge and releasing the phosphate group. The cross bridge is formed while the ATP is still attached. The ADP is then released and the myosin head slides the thin filaments past the thick filaments. This is called the power stroke. After the power stroke is completed, the cross bridge will stay intact until the myosin head binds another molecule of ATP, causing it to detach from the actin active site and go back into the low energy state. The calcium will then be pumped back into the sarcoplasmic reticulum, where the calsequestrin is. This will allow the tropomyosin to cover the actin active sites again, so the muscle may relax. The basic functional units of a myofibril are compartments called sarcomeres. The components of a sarcomere include Z-discs, which are narrow plate-shaped regions of dense material that separate one sarcomere from the next. A Z-disc passes through the middle of each I-band. An I-band is the lighter area that contains only thin filaments. It is also less dense. The A-band is the dark middle area of the sarcomere that extends the entire length of the thick filaments. It also includes the part where the thin filaments overlap the thick filaments. The H-band is the small area in the center of the A-band.